Ever wondered how your brain seamlessly orchestrates every single step you take, every gesture, even every tiny blink of your eye? It's far more complex and, frankly, elegant than you might imagine. Today, we're taking a deep dive into the intricate organization of motor function based on, well, a really rich physiological text. Our mission to pull out the most important nuggets of knowledge, give you a shortcut to being truly well informed about this incredible neuromachinery that gives us control over our bodies.
Yeah, it's truly fascinating when you stop and think about how much of our central nervous system is actually dedicated just to motor control. And understanding it is so crucial, not just out of curiosity, but because even small disruptions can cause significant motor symptoms. They're often vital diagnostic clues in the clinic. It's the blueprint for everything we do physically.
Okay, let's unpack this then. It sounds massive. With such a vast and fascinating system, where do we even begin? Maybe let's start right at the foundation, the spinal cord. You called it the central highway for movement commands. So what's its basic layout? And what exactly are motor neurons, these fundamental units?
Okay, yeah, the spinal cord. So the gray matter inside, it has that distinctive H or maybe a butterfly shape, depending on how you look at it.
Oh.
And tucked away in the ventral horn, that's the front part of the H. That's where you find the motor neurons. These are often called the final common pathway. It's a good term because they're the very last stop for any command coming from your brain or elsewhere in the nervous system before it actually reaches and activates a muscle.
Right. And within those motor neurons, I understand there are two key players, alpha and gamma motor neurons. How are they different? How do they talk to your muscles differently?
That's right. So alpha motor neurons, these are the big ones, large multipolar cells. Their axons stretch out from the spinal cord, they branch out, and they innervate multiple extrafusal muscle fibers within a single muscle. Now, these extrafusal fibers, those are the main workhorses, the big fibers making up the bulk of your skeletal muscle, generating the actual contractile force. An alpha motor neuron, plus all the muscle fibers it supplies, that forms what we call a motor unit. It's the basic functional unit of movement. And what's interesting is that each extrafusal muscle fiber gets input from only one alpha motor neuron.
Only one.
Yeah. And the size of these motor units varies wildly. Think about it. For incredibly precise muscles, like in your eye, an alpha motor neuron might control just a handful of fibers. But then you look at a powerful limb muscle like your quadriceps, and it can control thousands. Big difference.
That brings up a great point. How does your brain manage to generate such a huge range of forces? From picking up something delicate like a feather to lifting something incredibly heavy and do it with precision. It can't just be an on-off switch, right?
Exactly. Not at all. This is where the size principle comes in. And honestly, it's a really elegant solution. It's all about orderly recruitment, whether you're making a voluntary move or it's a reflex kicking in. The very first motor units activated are always the ones with the smallest axons. These connect to slow twitch fibers. They generate low force, but they're incredibly resistant to fatigue. Then, as your brain decides it needs more force, it progressively recruits larger and larger motor units. These bigger units have fast twitch fibers, capable of much higher force. So think of it less like an on-off switch and more like a finely tuned dimmer switch for muscle strength. Allows for incredibly smooth, graded, and efficient control.
Okay, so the alphas handle the main force? What about their smaller cousins, the gamma motor neurons? What's their specific job?
Right, the gammas. They are indeed smaller. And they don't directly control those main extrafusal muscle fibers. Instead, they synapse onto specialized intrafusal muscle fibers. These intrafusal fibers are part of tiny little spindle-shaped receptors called muscle spindles, which are embedded right within your larger skeletal muscles. So the gamma motor neuron's main job is basically to regulate the sensitivity of these muscle spindles, almost like keeping them perfectly tuned.
So it's not just the types of neurons, but also where they are in the spinal cord that matters. You mentioned a clear topographic organization in that ventral horn.
Absolutely critical. Yeah, the motor neurons in the ventral horn are very spatially organized. It's not random at all. A group of alpha motor neurons that innervates one specific muscle that forms what's called a motor nucleus. And the key thing is that motor neurons controlling muscles closer to your body's center. Your proximal muscles, like shoulders, hips, they're located more medially towards the middle, while those for your distal muscles, hands, feet, fingers, are located more laterally out to the sides. Also, neurons for flexor muscles tend to be positioned dorsal or behind those for extensors. And even the inner neurons, the connector neurons, they show a similar layout. This allows for both independent limb control, where lateral inner neurons project just to the same side, and also for bilateral coordination of your axial muscles, like in your trunk, where medial interneurons project to both sides.
Fascinating. So, for you listening, what's the core takeaway here? How does the spinal cord really set up this fundamental framework for movement?
I'd say the key is that it establishes the final output pathway, the motor neurons, and organizes them geographically, based on the muscles they control. And it sets up these basic units, the motor units, and the principle for how they're recruited for graded force, the size principle. It's the essential wiring diagram.
Okay, from individual muscle twitches to incredibly coordinated movements, it's clear our nervous system is a master orchestrator. You mentioned individual motor neurons control single muscles, but normal movement needs groups working together: agonists, synergists, antagonists, fixators. So how does the spinal cord actually link all these different activities together into a coherent whole?
Yeah, this is where reflexes really shine a light on the underlying circuitry. A reflex, by definition, is a fast, predictable, involuntary, and pretty stereotyped response to a specific stimulus. And because they're so predictable, spinal reflexes are incredibly useful tools for clinicians and for scientists to understand how the nervous system is functioning or maybe malfunctioning. The basic circuit is the reflex arc. It always has an offerent or sensory part, a central processing part, synapses, interneurons in the cord, and an efferent or motor neuron part that causes the response. We should probably explore maybe 3 key ones.
Okay, let's start with the most famous one, right? The myotatic or stretch reflex. Everyone knows the knee-jerk reflex. What is it, and what's the muscle spindle's role here?
Right, the knee jerk. So the stretch reflex is basically a group of motor responses triggered when a muscle gets stretched. The sensor, the receptor for this, is that muscle spindle we mentioned earlier. These are those spindle-shaped organs with the specialized intrafusal muscle fibers, and they've got tons of sensory and motor axons connected. Crucially, they lie in parallel with your main extrafusal muscle fibers. So when your main muscle stretches or shortens, the spindles do too, right alongside them. And they're particularly concentrated in muscles needing fine motor control, think hands, eyes.
And these spindles have different sensory fibers inside, group IA and group 2. How do they tell the difference between, say, how much the muscle stretched versus how fast?
Good question. The group IFPF fibers, they form what are called primary endings on the intrafusal fibers. These are super sensitive to both the amount of stretch and, really importantly, the rate of change, how fast it's stretching. The group 2 fibers form secondary endings, and they mainly respond just to the amount of stretch they give, more of a static, sustained response. It's a clever way for the nervous system to get different kinds of information from the same little sensor. Recent work points to the piezo2 channel as a key transducer here.
Okay, so this brings us back to those gamma motor neurons again. You said their activity is incredibly important for setting the sensitivity of these spindles. What does that actually mean for how the system works day to day?
Right, it's absolutely critical. Imagine if your main muscle shortened, contracted. The muscle spindle lying in parallel would go slack, wouldn't it? If it's slack, it can't detect any further changes in length effectively. It's like trying to detect a fish bite on a loose fishing line. This is where gamma motor neurons are essential. When your brain sends a command for the main muscle, via alpha neurons, to shorten, it often sends a simultaneous command to the gamma motor neurons. We call this alpha-gamma coactivation. This makes those little intrafusal fibers inside the spindle shorten right along with the main muscle. This keeps tension on the spindle's sensitive middle region. It ensures the muscle spindle stays sensitive, stays taut, ready to detect any further, maybe unexpected, small changes in muscle length, no matter how long or short the main muscle is.
Okay. That makes the stretch reflex itself incredibly powerful then. That quick knee jerk, the phasic part, is because those group IR efferents directly excite the alpha motor neurons going back to the same muscle.
Yeah.
Simple and fast. But there's also this concept called reciprocal innervation. What's that about? Sounds important for coordination.
It's absolutely crucial for smooth movement. So other branches of those same group IA fibers connect to special interneurons called reciprocal IA inhibitory interneurons. These inhibitory interneurons then they inhibit the alpha motor neurons that supply the antagonist muscles. So in the knee-jerk example, while your quadriceps, the extensor, is told to contract, these interneurons tell your hamstrings, the flexors, to relax. This organization, reciprocal innervation, ensures that when one muscle group is activated, the opposing group is simultaneously inhibited. It prevents you from fighting yourself, basically. Allows for fluid movement. And there's also a tonic stretch reflex, a slower response, which is really important for maintaining muscle tone and your posture. It's interesting, in the clinic, when those descending motor pathways from the brain are damaged, say, after a spinal cord injury, you often see these hyperactive stretch reflexes. This can lead to things like tremors or clonus, which is like a rhythmic muscle spasm. And it raises an interesting question. How can a reflex that's normally stabilizing suddenly become a source of instability? It's likely because the slight delay in the reflex loop, combined with an increased gain or sensitivity after injury, can lead to overcorrection in these unwanted oscillations. The damping mechanism is lost.
Wow, yeah, that's a great insight into how these systems can go wrong. Okay, so if the stretch reflex is mainly about muscle length, how does the inverse myotatic reflex, the Golgi tendon reflex, complement it? What's its focus?
The Golgi tendon organs, or GTOs, are the receptors here. They're found right at the junction where your muscle fibers meet the tendon. And the key difference, they lie in series with the muscle fibers, not in parallel like the spindles. Their job is to sense the tension or the force that's developing in the muscle or tendon. Doesn't matter if it's from passive stretch or active contraction, they sense force. They're innervated by group Ibert fibers.
And how does that reflex arc work? And you mentioned earlier they can act synergistically with the stretch reflex, even though they seem to do opposite things initially. How does that work?
All right, so the group EB afferents from the GTOs, they synapse on interneurons in the spinal cord. This leads to dysynaptic inhibition, meaning there's an interneuron in between of the alpha motor neurons going back to the same homonymous muscle. So it's basically saying, hey, tension is getting high, maybe back off a bit. At the same time, it often excites the alpha motor neurons going to the antagonist muscle. Now, while that sounds opposite to the stretch reflex, especially during passive stretch, think about standing. If your knee extensors start to fatigue, the force they generate drops. This reduces GTO activity. Reduced GTO activity means less inhibition of the alpha motor neurons to those extensors, which helps restore the force. It's disinhibition. Simultaneously, if your knee starts to bend slightly because of fatigue, that stretches the quadriceps, activating the muscle spindles, and those spindles excite the same alpha motor neurons. So you see, both reflexes can work together to help you maintain posture and prevent collapse or injury from too much force.
Okay, that clarifies the synergy. Now let's talk about withdrawal, the flexion reflex, and the crossed extension reflex, that immediate ouch, pull away response. How did that mechanism play out?
This is your body's main protective reflex, typically triggered by noxious or potentially damaging stimulus. Stepping on a sharp object is the classic example. Through a pathway involving multiple synapses, it's polysynaptic, it causes strong excitation of flexor muscles and inhibition of extensor muscles in the same limb, the ipsilateral limb. That's the withdrawal. But here's the really coordinated part. Simultaneously, special commissural interneurons cross the midline of the spinal cord, and they evoke the opposite pattern of activity in the contralateral limb excitation of extensors, inhibition of flexors. This is the cross-extension reflex. It helps you maintain balance by stiffening the other leg as you quickly pull the injured one away. Think about it, you'd fall over otherwise.
And what's truly fascinating, you mentioned earlier, is that the very interneurons involved in these flexion reflexes are also part of something called a central pattern generator, or CPG, for locomotion, like walking.
Exactly. It's a fantastic example of neural economy, how basic reflex circuits are reused and integrated into more complex rhythmic motor acts. These CPGs, located within the spinal cord, can actually generate the basic rhythm and pattern of walking, even without continuous sensory input from the legs or commands from the brain, though those inputs are crucial for adaptation and control. In fact, stimulating those same afferent fibers that trigger the flexion reflex can even reset or cause a phase shift in an ongoing locomotor rhythm. This shows how sensory feedback dynamically adapts these CPGs to the environment, like adjusting your step when you hit uneven ground. And just another clinical tie-in. After damage to descending motor pathways, those hyperactive stretch reflexes can lead to spasticity, a strong resistance to passive limb movement. Sometimes you see the clasp knife reflex where initial high resistance suddenly melts away. It was once thought to be solely due to GTOs, but it's now believed to involve other high-threshold muscle receptors too, reflecting the altered state of the spinal circuits when brain control is impaired.
Okay, we've laid some amazing groundwork with the spinal cord and its reflexes. But obviously, our movements aren't just reflexes we decide to move. So how do the higher brain centers initiate, refine, and orchestrate our voluntary and sometimes semi-voluntary movements? Let's get into those descending pathways.
Right, moving up from the spinal cord. Traditionally, people talked about pyramidal and extrapyramidal pathways. That terminology is a bit outdated now. A more functional way to think about them is classifying them into lateral and medial descending systems. These pathways mostly target interneurons in the spinal cord, though some, especially from the cortex, do make direct connections onto motor neurons. The lateral system primarily influences motor neurons on the opposite side of the body, contralateral, that control fine, skilled movements of your distal limbs, think fingers, hands, and also certain facial and tongue muscles. The key component here is the lateral corticospinal tract. It's quantitatively and functionally extremely important to humans, absolutely critical for that independent finger control we rely on so heavily.
Okay, lateral for fine control. What about the medial system?
The medial system is more concerned with posture, balance, and providing the sort of general motor background or supportive framework for your limb and digit movements. It tends to end on the medial groups of interneurons in the spinal cord and similar neurons in the brainstem. These often connect bilaterally to both sides, affecting axial musculature, your trunk, neck, shoulders. Key pathways here include the ventral corticospinal tract, the pontine and medullary reticulospinal tracts, the lateral and medial vestibulo-spinal tracts, crucial for balance responses, and the tectospinal tract, which helps coordinate head movements with visual and auditory stimuli.
And you also mentioned monominergic pathways earlier, descending from the brainstem. What's their specific role in this motor control picture?
Yeah, these are a bit different. They're less specifically organized in terms of targeting particular muscles. Think of them more as neuromodulators. Systems using serotonin or norepinephrine, for example, descend from the brainstem and basically alter the overall responsiveness and excitability of the spinal cord circuits. They don't usually initiate specific movements themselves, but they can kind of set the stage, adjusting the gain or readiness of the spinal cord for action.
Got it. Now, this is where the clinical relevance really hits home. Damage to these different descending pathways leads to very distinct patterns of motor problems. Let's take upper motor neuron disease, like after a stroke affecting the internal capsule where many pathways run. What symptoms do we typically see?
Right. In that classic scenario, you'll often see increased stretch reflexes, both phasic and tonic. That's the spasticity we talked about. You'll also see weakness or paresis, which is often most pronounced in the distal muscles like the fingers and hand. And you might see pathological reflexes pop up, like the Babinski sign, where stroking the sole of the foot causes the big toe to extend upwards instead of curl downwards. Interestingly, though, if you have a lesion that only interrupts the corticospinal tract itself, maybe in the medullary pyramid, spasticity might actually be absent or much less severe. You'd still see the distal weakness and the Babinski sign. This tells us that spasticity likely involves the disruption of other descending pathways too, like the reticulospinal tracts, probably due to the loss of that regulatory cortical influence over them. It's a complex interaction.
And if the medial system gets interrupted instead, what kind of problems arise then?
Quite different problems. Initially, damage to the medial system often leads to a reduction in postural muscle tone hypertonia and a loss of those automatic righting reflexes that help you stay upright if you're tilted or pushed. Long-term, the major issues tend to be locomotor impairment, difficulty walking smoothly, and frequent falling. But interestingly, fine manual dexterity, manipulating objects with the hands, might remain relatively normal. It really highlights the functional separation of these systems. This brings up the phenomenon of decerebrate rigidity. It's a specific state you can see after a particular type of brainstem injury, usually a transection in the midbrain. It leads to the state of exaggerated standing, with extreme hypertonia, especially in extensor muscles. How does that happen? It's because certain descending pathways from the cerebral cortex are cut off, but key brainstem pathways like the vestibulo-spinal and reticulospinal tracts remain intact below the lesion. The loss of cortical control dramatically alters the balance of activity in these intact brainstem pathways. You get increased excitation from pathways like the pontine reticulospinal tract, and maybe reduced inhibition from others, like the medullary reticulospinal tract. This imbalance leads to massive overactivity targeting motor neurons, causing the rigidity. And crucially, this rigidity can often be reduced by cutting the sensory nerve roots, dorsal roots, which tells us it heavily involves hyperactivity of the gamma motor neurons and the muscle spindle system. The lateral vestibulo-spinal tract also contributes by acting more directly on alpha motor neurons.
Okay, let's move up to the highest level, the true command center for voluntary movement, the cerebral cortex. How does the cortex plan and execute these complex, goal-directed movements? Movements that often vary each time we do them and seem initiated purely by thought or intention.
It's a huge computational challenge for the brain. Think about just reaching for a cup of coffee. Your brain has to first identify the target, the cup, then locate it in space relative to your body. Then it has to figure out a path, a trajectory for your hand and arm. And finally, it needs to compute the precise sequence and magnitude of muscle forces required to actually move your arm along that path smoothly and accurately. This involves a whole series of complex transformations between different coordinate systems, from where the image falls on your retina, retinotopic, to where the cup is in the room, external space, to coordinates relative to your body, body-centered, and ultimately down to the specific muscles involved, muscle-based coordinates. It's quite amazing it happens so fast.
And we know there isn't just one motor area in the cortex, right? There's the primary motor cortex, where movements need the least stimulation, but also the premotor area, the supplementary motor area, or SMA, even areas in the cingulate cortex, and the frontal eye fields for eyes, of course. Now, traditionally, these were seen as working in a strict hierarchy. Sensory info comes in, planning happens in premotor SMA, execution command goes out from primary motor cortex. But you mentioned earlier this idea of a parallel distributed network is more current. What does that mean practically?
Yeah, the strict hierarchy model is probably too simple. The newer view emphasizes that it's much more collaborative. All these motor-related cortical areas are heavily interconnected, with signals flowing back and forth constantly. They all contribute significantly to the descending pathways, not just the primary motor cortex. It's less like an assembly line and more like a highly interactive committee working together. For instance, the primary motor cortex itself only contributes maybe about half the fibers in the corticospinal tract that originate from the frontal lobe. Other areas contribute significantly too. Within the primary motor cortex, yes, there's that famous topographic map, the motor homunculus, where body parts are laid out. Larger areas represent parts with finer control like hands and face, but when you look closely with microelectrodes, it's more complex. It's not neat borders. It's more like an intermingled representation where cell columns controlling different joints are kind of interspersed. And what's truly remarkable here, maybe even more than the map itself, is that this map is incredibly dynamic. It's not static. It's constantly changing. Axons from neurons spread out, linking different cell columns. And the functional strength of these connections can be rapidly modulated on a millisecond timescale by local inhibitory interneurons using GABA. So, for example, if you were to experimentally block this local inhibition, the map can change dramatically. Stimulating one spot might suddenly evoke movements in muscles controlled by a normally distant region. It shows these latent connections are there. And of course, longer-term plastic changes happen too, based on experience. Use it or lose it applies here. Practice can expand a representation. Disuse can shrink it. It's incredibly adaptable.
So how do the individual neurons within the motor cortex actually contribute to planning and execution? Are they just firing like simple switches when a muscle moves?
No, it's much more sophisticated than that. We see that motor cortex neurons change their firing rates before the movement even starts. And the timing of this change correlates with your reaction time. Their firing rate often seems to correlate better with things like the amount of contractile force being generated by the muscle, or the rate of change in force, rather than just the final joint position. And some neurons show what's called set-related activity. They become active during a delay period when a movement is being planned or prepared but hasn't been initiated yet, maybe even when it's withheld. This strongly suggests they're involved in the planning and preparation stages, not just the final execution command. Now, an individual neuron might be broadly tuned. That means it fires most strongly for a movement in a particular preferred direction, but it'll still fire just less for movements in other nearby directions. So how do you get precise movement from these broadly tuned cells? The key seems to be looking at a population activity. Researchers developed this idea of a population vector. You represent each active neuron's contribution as a vector. The direction points in the neuron's preferred direction, and the length, magnitude, represents its firing rate at that moment. If you then sum up these individual vectors from hundreds of neurons across the motor cortex, the resulting overall population vector actually predicts the direction of the upcoming movement with remarkable accuracy. It's like the collective decision of the neural committee points the way. And importantly, neurons in the premotor and supplementary motor areas also show this kind of activity related to upcoming movement, correlated with parameters like force or target location. This really supports that distributed network view. Quantitatively, these areas might even have a higher proportion of neurons involved in those earlier motor planning stages compared to the primary motor cortex. Broadly speaking, the premotor area seems more involved when movements are guided by external cues like reaching for a visible target. The SMA seems more active for self-initiated movements, sequences, or movements guided by internal cues like playing a memorized piano piece.
Okay, fascinating. Let's pivot now to another major player, the little brain, the cerebellum. We know damage here doesn't cause paralysis, but it messes up coordination pretty badly. So what exactly is the cerebellum doing? What's its unique contribution to this whole motor control system?
Right, the cerebellum is a great coordinator. Interestingly, damage to one side of the cerebellum affects motor function on the same side of the body, the ipsilateral side. This is because its output pathways cross over twice before influencing motor neurons down in the spinal cord. Cerebellar damage typically leads to a trio of problems: issues with motor coordination, equilibrium or balance, and muscle tone. Clinically, this manifests in several ways. You see ataxia, which is that awkward, unsteady, wide-based gait. You see dysmetria errors in judging distance or force, leading to overshooting or undershooting a target, like when reaching for something. There's also dysdiadochokinesia, that's a mouthful, but it means difficulty performing rapid alternating movements, like tapping your fingers or flipping your hand back and forth quickly. Movements can become decomposed, losing their smoothness and breaking down into a series of discrete, jerky steps. An intention tremor is also characteristic of tremor that gets worse as you approach a target. Plus, impaired balance, a tendency to fall towards the affected side, and often hypertonia, or decreased muscle tone. You might see a pendular knee jerk, where the lower leg swings back and forth more than usual. EMG studies confirm the underlying problem often involves abnormal timing and patterning of agonist and antagonist muscle contractions during movement. It really messes up the fine-tuning.
And structurally, the cerebellum is quite distinct, sitting there in the back, connected to the brainstem by thick bundles of fibers called peduncles. It has an outer cortex, white matter tracts underneath, and these deep cerebellar nuclei buried within. And the cortex itself is folded and subdivided into lobes and longitudinal zones.
Exactly. And this intricate structure is absolutely key to its function. The cerebellum receives input from two main, very different types of afferent systems: mossy fibers and the olivocerebellar, or climbing fibers. Mossy fibers are excitatory. They come from a huge variety of sources: spinal cord carrying sensory info, brainstem nuclei, and even indirectly from the cerebral cortex via relays in the pons. They bring in a massive amount of information about the body's state and the external world, extraceptive and proprioceptive signals, and they actually form rough somatotopic maps within the cerebellum. These mossy fibers excite tiny granule cells, the most numerous neurons in the entire brain, whose axons then form the parallel fibers. These run perpendicular across the dendritic trees of the Purkinje cells, creating this incredible matrix, allowing for massive convergence and divergence of information. Input arriving via mossy fibers leads to what are called simple spikes in the Purkinje cells. These are relatively fast, continuous firing patterns, maybe hundreds of spikes per second.
Okay, so mossy fibers bring in lots of general information leading to simple spikes. What about the climbing fibers? They sound different.
Very different and quite unique. All climbing fibers originate from just one nucleus in the brainstem, the inferior olivary nucleus. This nucleus itself is unusual because its neurons have a lot of electrical coupling through gap junctions, allowing them to fire in remarkable synchrony. Now here's the striking thing. Each Purkinje cell receives incredibly powerful input from only one climbing fiber, but that one connection is massive, wrapping around the Purkinje cell dendrites. And when that single climbing fiber fires, it produces a huge depolarization in the Purkinje cell, causing a high-frequency burst of action potentials called a complex spike. These complex spikes are much rarer than simple spikes, maybe only not firing once per second or so. But when they occur, they briefly dominate the Purkinje cell's activity, reflecting that synchronized state of the inferior olive.
And what's truly remarkable, as you pointed out, is that these Purkinje cells are the only output from the entire cerebellar cortex. And they are they release GABA onto the deep cerebellar nuclei. So all this complex processing in the cortex ultimately results in an inhibitory signal shaping the output.
Precisely. It's this inhibitory output that sculpts the final command going out from the deep cerebellar nuclei. Now, what do these two different spike types, simple and complex, mean for function? That's been a huge area of research and debate. While simple spikes seem to reflect the ongoing processing of general sensory and motor information, those rare but powerful complex spikes are thought to be really significant. If we try to connect this to the bigger picture, one very influential hypothesis linked to motor learning is that complex spikes act as an error signal. The idea is that when a movement deviates from what was intended, the climbing fiber fires, triggering the complex spike. This then induces a long-term change, specifically long-term depression, LTD, at the parallel fiber synapses onto that Purkinje cell that were active just before the error. So it weakens the connections that contributed to the mistake, allowing the cerebellum to refine motor skills over time, basically. The neural basis of practice makes perfect. However, there's an alternative view, or maybe complementary view, suggesting the olivocerebellar system is also directly involved in real-time motor control, perhaps in timing motor commands by generating these precisely synchronized discharges that can effectively gate or pace the activity of the deep nuclear cells. So it raises this important question: is the climbing fiber system primarily for learning, or is it also crucial for the moment-to-moment execution and timing of movements? The jury might still be out, Or perhaps it does both.
Okay, another critical set of structures now, the basal ganglia. These are another collection of deep brain structures. We know they're vital for regulating motor activity, but also involved in things like habits, emotions, and cognitive functions. How do they work in the motor realm, and what happens when their delicate balance is thrown off?
Right, the basal ganglia. If the cerebellum is the coordinator, you can kind of think of the basal ganglia as the gatekeeper, or maybe a filter for movement. They play a crucial role in selecting desired movements and suppressing unwanted ones, ensuring our actions are smooth, purposeful, and appropriate to the context. The key motor components include the dorsal striatum, which is the caudate nucleus, and putamen, the globus pallidus, with its external GPE and internal GPI segments, the substantia nigra, pars compacta, and pars verticulata, and the subthalamic nucleus. They don't project directly down to the spinal cord. Instead, they form these complex loops. They receive massive input from almost the entire cerebral cortex, primarily into the striatum. Then they process this information through internal circuits, and their main output, largely from the GPI and substantia nigra pars reticulata, goes to the thalamus, specifically the ventral anterior VA and ventral lateral VL nuclei. And these thalamic nuclei then project back up to the motor areas of the cortex, the premotor, supplementary motor, and primary motor cortex. So it's a crucial corticostriato-thalamocortical loop.
And central to understanding how this loop works are the concepts of the direct and indirect pathways through the basal ganglia. They seem to have opposing effects on movement. Can you walk us through the direct pathway first, sometimes called the GO pathway?
Absolutely. The direct pathway's overall action is generally thought to facilitate movement, to release the brakes. It starts with excitatory input from the cortex to specific neurons in the striatum. These striatal neurons then send inhibitory signals directly to neurons in the GPI, internal globus pallidus. Now, the GPI neurons are themselves inhibitory, and they are tonically active, constantly putting a brake on the VA-VL nuclei of the thalamus. So when the direct pathway inhibits these GPI neurons, it's like inhibiting an inhibitor, which results in disinhibition of the thalamic neurons. The brake is released. These disinhibited thalamic neurons can then increase their firing rates, sending stronger excitatory signals back up to the motor cortex, thereby promoting or facilitating the initiation and execution of movement.
Okay, so direct pathway releases the brake. Then the indirect pathway must be the opposite, the no-go or stop pathway, applying the brake.
Exactly. The indirect pathway's overall effect is generally to suppress or reduce motor activity. It also starts with cortical excitation, but to a different population of striatal neurons. These neurons then inhibit the GPE, the external globus pallidus. The GPE neurons normally provide tonic inhibition to the subthalamic nucleus, so inhibiting GPE leads to disinhibition of the subthalamic nucleus, releasing the brake on it. The now disinhibited subthalamic nucleus neurons become more active, and they send excitatory signals to the GPI. This increases the activity of those inhibitory GPI neurons. More GPI activity means greater inhibition of the VA-VL thalamic nuclei. This increased braking action on the thalamus leads to decreased excitatory drive back to the motor cortex, ultimately suppressing or reducing movement. And mediating this whole balance critically is dopamine, released from neurons originating in the substantia nigra pars compacta, SNPC, that project widely within the striatum, the nigrostriatal pathway. Dopamine has this really clever dual effect that ultimately facilitates movement by tipping the balance towards the direct pathway. It excites the striatal neurons of the direct pathway, acting via D1 receptors, and inhibits the striatal neurons of the indirect pathway, acting via D2 receptors. So dopamine essentially hits the accelerator on the go pathway and hits the brake on the stop pathway. This delicate balance is crucial for normal, fluid movement. When it gets disrupted, you see profound motor disorders.
And this is where it gets really interesting and clinically very relevant. Imbalances between these direct and indirect pathways lead to very distinct movement disorders, often affecting the opposite side of the body from where the damage is in the basal ganglia. Let's start with Parkinson's disease.
Parkinson's is the classic example of a hypokinetic disorder, too little movement. It's characterized primarily by tremor, often that tool rolling tremor at rest, around 3 to 5 hertz. Rigidity, where the limbs feel stiff like a cogwheel when moved passively, and bradykinesia, which is a marked slowness in initiating and carrying out movements, sometimes even progressing to akinesia, a lack of movement. The underlying cause is the progressive degeneration of those dopamine-producing neurons in the substantia nigra pars compacta. With the loss of dopamine's modulatory influence, activity in the direct pathway is diminished, and activity in the indirect stop pathway is increased. The net result is that the GPI becomes overactive, putting excessive braking force on the thalamus. This leads to reduced activation of the motor cortex, causing the characteristic difficulty initiating movement and the overall slowness. Therapies like levodopa, which is a precursor to dopamine, or deep brain stimulation, DBS, often targeting the subthalamic nucleus for GPI, aim to try and restore this balance within the loop.
Okay, Parkinson's is too little movement. Then there's Huntington's disease, which presents very differently, with too much movement, right?
Exactly. Huntington's is a prime example of a hyperkinetic disorder. It's an inherited genetic disorder, autosomal dominant. It primarily involves the progressive degeneration of striatal neurons, particularly, at least early on, those belonging to the indirect pathway. Later, neurons in the direct pathway and the cortex itself are also affected. With the loss of those indirect pathway neurons that normally inhibit the GPB becomes less inhibited, leading to more inhibition of the subthalamic nucleus. This reduces the excitatory drive from the subthalamic nucleus to the GPI, so the GPI becomes less active. Less GPI activity means less inhibition or disinhibition of the AVL thalamic nuclei. The brake is effectively removed. This leads to excessive activation of the motor cortex, resulting in the characteristic hyperkinetic symptoms like chorea, those rapid dance-like involuntary movements, and athetosis, which are slower, writhing, twisting movements. The gait is stuck open.
And finally, Ballism is another, perhaps even more dramatic, hyperkinetic disorder linked to the basal ganglia.
Yes, Ballism, or often hemiballismus if it affects one side of the body, is characterized by really uncontrolled, violent, flinging movements of the limbs. It's typically caused by damage, often a stroke, specifically within the subthalamic nucleus. Remember, the subthalamic nucleus normally excites the GTI, so if you lesion the subthalamic nucleus, you lose that excitatory drive onto the GPI. GPA activity plummets, leading to drastically reduced inhibition of the thalamus. This unleashes the motor cortex, causing those wild, large amplitude involuntary movements. It really underscores the critical role of each node in this intricate circuit.
Okay, we've covered limb and body movement control extensively, but our eyes have their own incredible, specialized motor control system. It seems simpler in terms of just a few muscle pairs per eye, but the speed and precision required are astonishing. What stands out to you about why eye stability is just so absolutely crucial for vision?
That's a fantastic point, and it really is fundamental. Our visual acuity, our ability to see detail, drops off dramatically if the image of the world slips across the retina too quickly. So perhaps the most fundamental job of the eye movement system is to hold the visual image steady on the fovea, that tiny spot in the center of the retina, with the highest concentration of cones, and thus the sharpest vision, especially when our head or body is moving. To achieve this, and also to direct our gaze, we have several specialized types of eye movements, each with its own purpose and neural control system. First off, there's the vestibulo-ocular reflex, VOR. This is probably the primary mechanism for maintaining visual stability during head movements. It generates eye movements that are equal in speed and opposite in direction to head movements. Turn your head left, your eyes automatically move right, keeping your gaze fixed. It's initiated by signals from the vestibular system in your inner ear, the semicircular canals detecting rotation, the otoliths detecting linear motion and gravity. And remarkably, it works perfectly well, even in complete darkness. It's an incredibly fast, essentially open-loop reflex system, though if it fails, other systems can compensate. Second, we have the optokinetic reflex, OKR. This is another system for stabilizing the visual scene, often working hand-in-hand with the VOR, especially during sustained movements. The OKR is activated not by head movement directly, but by the movement of the visual scene itself across the retina, what's called retinal slip. Think about watching the scenery go by from a train window. Motion-sensitive cells in the retina detect this slip, and the reflex tries to move the eyes to minimize it, helping to keep the image stable, particularly for slower, prolonged head movements where the VOR might fade slightly. Then we have saccades. These aren't for stabilization, but for shifting gaze rapidly. They are very fast, jerky, ballistic eye movements that quickly redirect the fovea to a new object or point of interest in the visual field. When you read, for example, your eyes jump from word to word using a series of saccades. They are incredibly fast-reaching speeds, up to 800 degrees per second in humans. Saccades can be voluntary. You decide to look somewhere, or reflexive, triggered by a sudden stimulus in the periphery. An interesting phenomenon is saccadic suppression. Our visual perception, especially for motion, is actually dampened just before and during a saccade. This prevents us from perceiving a bloody streak every time our eyes jump. Control involves the superior colliculus in the brainstem and cortical areas like the frontal eye fields. Complementary to saccades are smooth pursuit movements. These are much slower, smoother, continuous eye movements used to track a moving target accurately, keeping its image right on the fovea. Think of watching a bird fly across the sky.
This requires predicting the target's motion and involves suppressing the OKR reflex that would otherwise try to stabilize the gaze on the background. It's a more complex process involving visual cortex areas, parts of the temporal lobe, the frontal eye fields, and critically the cerebellum for accuracy. We should also mention nystagmus. Technically, nystagmus is any repetitive, rhythmic, to and fro movement of the eyes. It usually has alternating slow and fast phases. Physiological nystagmus occurs naturally during prolonged VOR or OKR stimulation. For instance, if you spin in a chair and then stop, you'll experience post-rotatory nystagmus. Your eyes try to compensate for the perceived continued rotation, slow phase, then quickly reset back, fast phase, when they reach the edge of their range. Pathological nystagmus, however, can be a sign of underlying neurological or vestibular problems. For example, damage to the inner ear, labyrinth, or vestibular pathways can cause an imbalance, leading to spontaneous, persistent nystagmus even when the head is still. The direction and type of nystagmus can be a valuable diagnostic clue. The mnemonic WS cold, opposite warm same, helps clinicians remember the direction of the fast phase of nystagmus during caloric testing of the vestibular system. And finally, vergence movements. These are unique because the two eyes move in opposite directions. They converge, turn inwards, to look at a near object, and diverge, turn outwards, to look at a far object. These disjunctive movements are essential for maintaining binocular vision and ensuring the image of the target falls on both fovea simultaneously, preventing diplopia or double vision. They are primarily triggered by stimuli, like retinal disparity, the slight difference in the image seen by each eye, and blur, as objects move closer or farther away. The cerebellum also plays a significant role in coordinating vergence movements.
And the underlying neuromachinery, the circuitry for these movements, especially the motor neurons supplying the 6 extraocular muscles around each eye, is highly specialized, isn't it? Controlled by 3 cranial nerves, the oculomotor, trochlear, and abducens nerves. And they form some of the smallest motor units in the entire body, sometimes just 1 nerve fiber to maybe 10 muscle fibers. That points to incredibly fine control.
Absolutely. That extremely low innervation ratio is key to the precision needed. Most of these extraocular motor neurons are also tonically active, meaning they have baseline firing rate even when the eye is still. Their firing rate then increases or decreases precisely in relation to both the position and the velocity of the eye movement. This allows for a very finely graded push-pull action between agonist and antagonist muscle pairs to control eye position and movement with exquisite accuracy. And the coordination between different muscles and between the two eyes relies on intricate brainstem pathways, like the medial longitudinal fasciculus, MLF, which links the different cranial nerve nuclei involved in eye movements. The VOR circuitry is a beautiful example of this precision working automatically. Let's take the horizontal VOR again. If your head starts rotating to the left, this stimulates hair cells in the left horizontal semicircular canal, increasing the firing rate of the left vestibular nerve afferents. These afferents then excite neurons in the contralateral right abducens nucleus. These abducens neurons do 2 things. They directly activate the right lateral rectus muscle, pulling the right eye outward rightward. And via special internuclear neurons that travel up the MLF, they also excite motor neurons in the left oculomotor nucleus that control the left medial rectus muscle, pulling the left eye inward rightward. At the very same time, inhibitory neurons project from the left vestibular nucleus to inhibit the ipsilateral left abducens nucleus, relaxing the left lateral rectus and right medial rectus. This whole 3-neuron arc ensures perfectly coordinated conjugate eye rotation to the right, exactly opposing the leftward head movement, all happening incredibly fast to keep your gaze stable. And again, the cerebellum constantly monitors and adjusts this reflex gain based on experience.
It's truly mind-boggling, just the sheer variety and precision of these different eye movements all working together seamlessly. For you listening, what's the one key takeaway? How utterly crucial is this specialized system to just navigating our daily lives?
I think the takeaway is that stable, accurate vision, which we completely take for granted, relies on this incredibly sophisticated, fast, and highly specialized motor system working perfectly in the background all the time. Without it, the world would just be a blur.
So we've taken quite a journey today. From the very fundamental motor units down in your spinal cord, up through the complex interplay of the cerebral cortex, the coordinating power of the cerebellum, the gating function of the basal ganglia, and culminating in the absolutely astonishing precision of our specialized eye movements. It really is a testament to the intricate, coordinated symphony that's happening constantly inside you, allowing you to interact with the world around you.
Absolutely. And maybe stepping back for a final thought. If we consider the incredible dynamic plasticity and the adaptive learning capabilities built into these neuromotor systems, we see it in how the brain can rewire its motor maps with practice or injury.
How the cerebellum refines movements leading to that practice makes perfect phenomena. How might our deepening understanding of these circuits, these blueprints for movement, actually redefine the future possibilities for rehabilitation after injury or disease? Could we potentially move beyond just recovering lost function? Could we perhaps even find ways to enhance our natural motor abilities, maybe pushing the boundaries of skill and performance in ways we can barely imagine today? Or even think about how this knowledge could lead to much more seamless and intuitive integration with advanced prosthetics or brain-machine interfaces, effectively creating entirely new ways for us to interact physically with the world. It opens up some really profound questions.
Definitely food for thought. So consider that next time you just casually pick up a cup or take a step or even just let your gaze drift across the room, it's not just a simple action. It's truly a marvel of biological engineering, a system constantly learning, adapting, and refining its incredibly precise blueprint, all happening right there inside your head.